is not part of mitosis, but it is part of the cell cycle. In fact, in this part, the cell is not dividing, it's just growing and preparing for the next cell division. The interphase has three parts, the G1, S, and G2. The G stands for gap. In these gaps, the cell is just growing and creating the materials and the organelles needed to mitosis to occur. But in the S part, which happens in between the gaps, the DNA is duplicated. That's why it's called the S part, because in this part, the synthesis is being made.
During prophase, inside the cell, the condensed materials and the duplicated chromosomes become visible. Also, during prophase, each DNA strand of the duplicated chromosome is referred as a chromatid. And outside the cell, the spindle fibers become deformed, and they are made up of microtubules, a kind of protein, and have a finite form. Also, the centers that were duplicated during interphase start to move towards opposite side of the cell. Prophase ends when the nucleolus disappears and the nuclear envelope breaks down.
The second stage of mitosis is metaphase. In this stage, the centromeres of the duplicated chromosomes line up across the cell.
The third phase of mitosis, anaphase, begins when sister chromatids separate and move apart. Once anaphase begins, each sister chromatid is now considered a single chromosome. During anaphase, chromosomes separate and move along spindle fibers to opposite sides of the cell. Anaphase comes to an end when this movement stops and chromosomes are completely separated into groups.
Telophase is the fourth and final phase of the mitosis. Chromosomes begin to spread out into a tangle of chromatin. The nuclear envelope reforms around each cluster of chromosomes. The spindle begins to break apart and the nucleus becomes visible.
Cytokinesis completes the process of cell division. It splits one cell into two.